Let's begin the next part of affinity chromatography, that is how to select a support matrix that we have talked about, right? So ideally talking, the support matrix characteristics and its activation with the ligand and the spacer arm concepts you have learned now. So when it comes to support matrix characteristics, the affinity purification, when we are talking, several factors are to be considered when you are choosing a support material. Of course, like any other uh, column chromatography matrices, it should be chemically inert, right? Good chemical stability it should have. Mechanical stability it should have. Its pore size and particle size also are going to be very important when we are talking about selecting the support matrix. So characteristics desired that of an ideal insoluble matrix for affinity chromatography, when we say, then we have to remember that the matrix should be inert to other molecules. So it is not going to have more interactions which are non-specific. So you have to have matrix that is going to be inert with respect to other molecules so that you minimize on non-specific adsorption. Now, secondly, pore size and particle size, when we say, the phenomena of uh, clogging of the column due to its own weight should be avoided. So the good flow rate is to be maintained. So the pore size, particle size, porosity, all these factors and the strength of the gel material are going to decide the flow properties. So the gel matrix for the affinity chromatography should have good flow properties. Secondly, it is very important that all these properties should not get altered due to the chemical environment that is present in the column during running the column or during elution of the column. That means it should be chemically and mechanically stable at different phases of the process, which are going to use different mobile phases, different buffers, elution buffer, for example. So the microenvironment is going to get altered. You are going to have different pH, different ionic strength, maybe denaturing conditions which are applied for binding and for elution. When you denaturing, as to not the denaturation of the molecule but denaturation of the interaction between the ligand and the analyte. So at all these conditions, your support matrix should be stable and it should be flexibly designed so that it remains working under such varying conditions and is stable. Third, next is like uh, large number of suitable chemical groups it should have, because when it is about the chemical groups, the chemical groups, as we have discussed in the previous lecture, these chemical groups are important for binding with the ligand. So the support matrix should have stable chemical groups, not only large numbers, but suitable chemical groups that would interact selectively with the support matrix, but or that of the support matrix will react with the ligand but will not have any interaction with the desired analyte. Further, it should be preferably highly porous. High porosity provides a large surface area for attachment of the ligand, and better interaction can be achieved and the desired molecule can be efficiently separated. So when it comes to practice of affinity chromatography, the particle size of course has to be uniform. The particles are to be spherical, they should be rigid so that good flow rate, good stability, high surface area can be achieved. So as we have talked earlier, agarose polyacrylamide gel, which are the controlled porosity we are going to get depending upon how they are chemically treated. So polymerization, these are the natural polymers. How do they look like, or how they are activated, that we have learned in the previous lecture. Again, I'm not going to repeat that. So maybe agarose or polyacrylamide gel, or maybe simple glass beads of defined porosity can be used. Most commonly, of course, agarose beads, most often they are used because they possessed all the basic features that we talked about, that of a good affinity support matrix. But the problem with agarose is that, which is you can say disadvantage of using agarose, is its susceptibility to different elution or denaturation conditions that are used to elute the analyte. So at the end of the chromatography, when you want to elute the desired analyte from it, the elution buffers or denaturation conditions which are used, under such conditions agarose is going to get affected, being a natural polymer. Of course, polyacrylamide beads, they are better, and they fulfill the desired criteria, but they also have a problem, that is the porosity is less as against that of agarose. So mm, when it is important to substitute it with ligands. So the controlled pore size glass beads, they are good with respect to mechanical rigidity. Totally chemically they are inert and provide a very good flow rate. So many a times glass beads also can be chosen. But however, sometimes non-specific protein adsorption is observed when it is glass bead that is used. So every kind of a support matrix, when you are talking about, it has its some good points, some limitations, and you have to select the support matrix very selectively. So this was about the porous supports, right? Now, other types of supports which are used include non-porous materials, membranes, flow-through beads like perfusion medias, right? So what are they? So to talk about the non-porous support matrices or materials which are used, they are of a size say about 1 to 3 microns. They give you fast purification, but again, the because the particle size is small, surface area that we talk about, which you obtain from non-porous materials, is limited as against that of porous materials. Again, membranes are talked about, which are good for affinity chromatography. They do not have pores, so the surface area is limited, but separation is better with membranous support matrices. The third type is flow through beads or perfusion media. They have both, that is large and small types of pores are present in it, and the flow rate can be adjusted and directly transported. Direct transport of the mobile phase in and across the beads is possible. Along with these beads, sometimes small analytes also can travel into it, so interior of the particle. But when you talk about direct transport to the interior, that means only short distances are required for diffusion, and diffusion is one of the basic inherent principle that works in any type of chromatography, right? Partitioning, that is what we had talked about, and diffusion is another thing that is very important when we talk about chromatographic separations. Now, considering the limitations of all these, do we have any other types of support matrices with the advancement in the technique? Monolithic supports. So when you talk about monolithic supports, their principle is similar to perfusion media. They have both large flow through pores and small through pores. So monolithic, you are going to have diffusion pores and they can be used. Even advanced version is expanded bed adsorbents. In this, as you are seeing in the diagram, the expanded bed adsorbents. Now here, if you observe the column, the first column is showing you bead particles which are separated from each other, and they are almost up to the complete height of the column, and this is the pressure pump, right? But in the second case, you are seeing all of them are compressed and the pressure pump is uh, pushed down, right? The uh, Now, when you talk about this, it is very important to see the horizontal lines which mark it, which tell you about the step in the affinity process, chromatography process. So adsorption and wash mode, you are going to have this kind of cross section of the column, and when it is elution mode, it is going to be of this side. So when you want to elute, you are going to compress the column, right? The bed which was expanded is to be compressed. And how this expansion happens? Its expansion happens due to reverse flow that is allowed. So the reverse flow is allowed during adsorption phase and washing mode. So when the column is allowed to develop, that time you are going to have expanded bed uh, uh, de- uh, status. But when you are going to elute, you are going to press it. So generally, as I said earlier, many a times when you work for high length or very long columns, and those with very small size particles, because smaller the particle, spherical size, better is the surface area, so separation is going to be better. But it is going to get affected from this the clogging problem. Its own pressure is going to clog its sites of action, and separation or partitioning will not be efficient. So this clogging problem can be avoided if we are taking help of expanded bed adsorbents. Now, going to the next part in it, when we talk about affinity chromatography purification, of course, like any other chromatography, it can be attached to the detection systems and analyzers, and what you get is tabular data as well as graphical data. But here, to take the cross section, how the entire system is going to function when samples are applied, uh, the conditions are to be such that maximum binding of the analyte occurs onto the column. Now here, if you see, on the x-axis you have column volume, right, and on the y-axis you have adsorbance of protein or the analyte of your interest, that is plotted. Now we are talking with respect to different phases. First is equilibration phase, when you are going to run down the mobile phase so that your column is going to get equilibrated at that particular microchemical or biochemical environment, right? So once your column is equilibrated, you are going to load your sample onto the column. When it is loaded, it is shown with this arrow. At the end of equilibration, you are going to load the column. That means your sample analyte is placed on the top of the column. Different methods, mechanism, instruments for loading the sample analyte we have discussed in the previous lectures, so I'm not going to repeat on it. Now, as sample starts running down the column, right, it will be interacting with the ligands and will start binding, right? And during this phase, only the unbound mm, uh, analyte components will come down from the mixture. You have different types. One is selective, which you want to retain, it will bind onto the column ligand, and rest all will come down along with the mobile phase that is running through. But of course, here, this is the major time that you have to allow your chromatographic column uh, working when you talk about. So when a sample is applied, after that a washing step is applied to remove unbound substances, and which is going to be the end part of it, right? So you are going to allow unwanted unbound materials to be washed away. Now, the case is that all your analyte of your interest has got adsorbed onto the ligand or the matrix or the activated matrix, whatever you call it, that of the column, right? And now it is to be eluted, for which you are going to use buffers. So here there are two different mechanisms which are used. Now elute the bound or adsorb analyte, you have to elute and then re-equilibrate the column so that you can reuse it. That is to be done. Now for elution, you can be using a specifically a competitive ligand which is put into the mobile phase. So there is attraction force stronger in the mobile phase, so your analytes gets dissolved from the support matrix ligand and will come down in the flow. Or secondly, you can alter the physicochemical environment, the microatmosphere, by changing the ionic strength or pH or polarity of the buffer, and then you can get its elution, right? Now, when we talk about efficiency of the column, we talk in terms of how selectively your analyte is getting separated from rest of the components in the mixture. You don't want any contaminant molecule remaining with your analyte, right? And you want maximum concentration of your analyte from the entire sample. So when you are talking about this, Very importantly, the type of ligand that you have chosen, the support matrix that you have chosen, at the same time, the spacer arm that you have chosen is going to play a major role. And that is, we have discussed it in the previous lecture today. But when it comes to elution and seeing the results, what kind of effect it will have when it is with reference to efficiency of the process. Now, uh, in the slide, you are seeing two different graphs. In the first graph, you are seeing this is the support matrix. This blue dot is nothing but the ligand, which is selective for your analyte, which is shown as a semicircle with a specific shape of site of interaction, like we show for lock and key mechanism of enzyme model. Similarly, right? So the ligand is attached onto the matrix, and as we run the column, the biomolecule of your interest will bind selectively to the ligand. Right? Now, same is going to happen in the case 2. That is, your analyte, while running down the column, is going to bind to the ligand. But because the ligand is spaced away from the matrix, binding of your analyte onto the ligand is much efficiently done. If you compare this part of the diagram with this, right, some of the sites are not on the analyte are not interacting well with the ligand. So anchorage of the analyte onto the ligand is loose, is weak. Here it is proper adsorption of analyte onto ligand is observed. So if this is the case, efficient binding is achieved, you can get better purification of your analyte. And this is just because presence of this spacer arm, which we talked about, we have amino, dipropyl, imino or nitro uh, kind of say spacer arm. So amino or imino group which is there on its either side of the rod, which is schematic representation for the spacer, which is one is going to react with the analyte, another is going to react with the support matrix, and in between you have hydrophobic group like propyl, butane, etc. Right? So this is going to push away the analyte from the support matrix surface, and so there is no hindrance, no steric hindrance. If there are no steric hindrances, you have efficient binding, and efficient binding will give you good separation of the analyte. Is it clear? Right, when we talk about these various ligands, selectivity and selection of the ligand and its properties that we talked in the previous slide also, I just go back to that. A ligand should have a strong interaction, but it should not be as strong as that it will require very drastic elution. Right, so when we talk about changing the elution buffer composition so that the ligand and analyte, those which are interacting, the interaction will weaken and the analyte will move down the column, will get eluted from the column in the elution buffer. But if this interaction between the ligand and the analyte is very strong, then very drastic uh, uh, what we say drastic elution conditions will be required. And if drastic conditions are to be implemented, then there is a danger of denaturation or loss of functionality, or the native structure of protein or biomolecule will get altered. So ligand should be strong, only interacting with the analyte, but at the same time it should not be a very strong interaction. Secondly, the ligand should have functional groups that can form the covalent bond with the support matrix and a good linkage with the support matrix. But when it is reinteracting with the support matrix, making a good linkage with support matrix at the same time, this linkage with the support should not interfere the interaction with the desired analyte. And there comes the purpose of the spacer arm and how it affects separation that we have talked about. Right, now let's take one example and then we conclude for today. That is, calcium dependent affinity ligands for purification of antibody fragments at neutral pH. right? Each word is very important. Calcium dependent, so calcium is going to work in the neutral and alkaline range. So neutral pH is very important when we talk about native and functional structure of biomolecules. Second is purification of antibody fraction. Generally, in the first lecture, when we talked about affinity chromatography, we talked about antigen antibody interactions as the sole basic principle of biological specificity is explored. Antibodies are coated onto the support matrix, and as the antigen mixture flows down, antigen will get selectively bound to the pre-adsorb antibody. But here, for example, monoclonal antibodies are used for purification. But here I'm not talking about the entire antibody, but I'm talking about the fraction of the antibody. What is that fraction? If you see the diagram carefully, or the title, it is SCFV, that is single chain variable fragment. You have variable fragments and fixed fragments. So out of that, single chain variable fragment that is used, that needs to be purified. Why to go with it? So when we talk about therapeutic proteins, that time this SCFV, single chain variable fragments of antibodies, have been found to be very important and advantageous over the conventional antibodies for therapeutic purpose. So when it comes to application for a tumor penetration, or when it comes to procedures like binding events in relation to injected mass, or uh, the size of the material matters, right? So as against the full length antibodies, these SCFVs are almost one fifth of the size, so the penetrability and functionality are better. So these SCFV are considered as the good therapeutic candidates, and their purification is done using chromatography, right? There are when we talk about the protocols which are going to be used for SCFV, that is single chain variable fragment of antibody, to be purified, you have to have selective elution under for each type of the antibody and its SCFV region, right? But many a times it is observed that it requires acidic pH, and that acidic pH is going to have effect on the proteinic nature of this antibody fragment, and aggregation of SCFE occurs, and the whole purpose of purification is compromised. So um, advancement in the process is use of calcium for better removal of this SCFE. So for example, when you're talking about uh, purification of the SCFEs, it is done with a calcium dependent manner. So they have developed specific ligands which are equipped with new and selective binding surfaces, and in which your calcium ions are used. So just with the change of pH, right? So if you take this diagram, you can see, right, that this is the chromatographic column, and it is the cross section of one of the beads. So in one of the beads, what you are seeing is, this is the linkers, right, or ligands, and this is the SCFV fraction, right, single chain variable element. So it is linked with SC and FV, and they are linked with the peptide segment, right, helical segment. Now here, this is uh, the buffer that is used is citrate buffer at pH 7. As this pH is changed to neutral, as you are saying, the adsorbed SCFA fraction is getting separated from it. So elution without aggregation of SCFA fragments is happening when we are going with a simple modification, that is calcium dependent affinity ligand for purification of antibody fragments, right? So when it comes to affinity chromatography, just to summarize today's um, discussion with reference to spacer arms, or that and ligands that we are talking about and their importance, there is one more thing that you have to take into consideration, on the basis of which affinity chromatography method is divided into two types. The first method is when you are using the structures or sequences of amino acids in the protein for binding site, right? So the examples include, for example, albumin bilirubin binding site, or binding of protein A in the support to the FC region of the IgG antibody, right? So these ligands are immobilized or coupled directly onto solid support matrices. So formation of the chemical strong bonds like covalent bonds between the particular functional group of the ligand, that of the support matrix, will give you a good activated matrix. So primary amines, sulfur hydryl groups, carboxylic acids, aldehydes, other reactive groups on the support, which are going to interact with these specific sequences or functional groups, which are the inherent components of the protein as the binding site, they are going to work. But in the second method, which is indirect coupling method, in that you are going to add a tag, and that is a specific amino acid sequence. So in the engineered protein, so that sequence you have purposely added to that, which is not having any function with respect to that functionality of the protein is concerned. However, it is just going to play a role as a tag. So number of different tags are available, and it could be as a simple sequence, amino acid sequence, as I said earlier, but it could be generally polyhistidine tag, which is commonly used. So it will selectively bind to the metal containing complexes and will purify that protein having that particular metal. The example which is purified very commonly, and the first one to be purified by this method, includes glutathione transferase sequence, which binds to glutathione, and it is going to have a selective tag which is going to interact with the enzyme selectively. So considering these things, calcium is going to be calcium binding site to a particular sequence, right, metal, in the ligand, and that with the analyte is explored in this case, and you are getting a better separation purification of SCFA fractions which are having therapeutic applications. Thank you.